Image like no other, inspiring decades of research and debate. The Shroud of Turin, that's a piece of history, and it has an ongoing puzzle for science.
The Shroud of Turin is a burial cloth that's imprinted with what many believe is the actual image of Jesus after his crucifixion. Now, it's been debated for years, but the shroud is back in the headlines in a major way.
For centuries, one mysterious piece of cloth has divided the world between faith and skepticism. Hidden within the ancient fibers of the Shroud of Turin lies the faint image of a crucified man, a figure millions believe to be Jesus Christ after the crucifixion. Some call it the greatest holy relic ever discovered. Others call it the most brilliant forgery in human history. But now, science has entered the debate. Deep inside the linen fibers, researchers uncovered traces of DNA, microscopic particles, blood residues, and chemical signatures unlike anything they expected to find. But before we reveal what the scientists discovered, take a moment to support the channel. Subscribe now and join us as we uncover history's greatest mysteries hidden beyond the reach of time. What began as a routine forensic investigation quickly turned into one of the most controversial scientific mysteries of the modern age. Geneticists, physicists, and forensic experts examined the shroud not as a sacred object, but as evidence, a silent witness carrying the scars of an event that may have happened 2,000 years ago. They scanned every thread, they analyzed every stain, and what they discovered shocked even the scientists themselves. The results challenged both skeptics and believers alike, because the shroud appeared to contain signs of torture so precise they defied medieval reproduction, traces pointing far beyond Europe, and evidence of a phenomenon modern physics still struggles to explain. Was the Shroud of Turin a masterpiece of deception? Or could it be the single greatest archaeological discovery in human history? Tonight, we follow the evidence hidden within the fabric itself. Through DNA analysis, forensic reconstruction, ancient pollen, blood chemistry, and unexplained energy patterns, to uncover the truth behind one of humanity's greatest mysteries. That historic moment arrived on May 28, 1898, in the city of Turin. A lawyer, local councilman, and enthusiastic amateur photographer named Secondo Pia received rare authorization from King Umberto I to photograph the mysterious relic during a public exhibition celebrating the royal wedding. At the end of the 19th century, photography was anything but easy. The process was slow, delicate, and highly technical. Pia carried an enormous camera into the cathedral, climbing high scaffolding, while working under dim lighting conditions. To capture the image, he depended on intense magnesium flashes and powerful lamps. Using two massive glass photographic plates measuring nearly 50 by 60 centimeters, he carefully took the photographs. Later that night, alone inside his darkroom, Pia began developing the plates under the dim red glow of a lantern. Then, something extraordinary happened. As the image slowly appeared in the chemicals, he was reportedly stunned to the core. Emerging from the negative was not the faint, faded outline visible on the cloth itself, but a remarkably clear and lifelike positive image of a man. A face with closed eyes, a swollen cheek, a damaged nose, a beard split at the center, features marked by severe suffering. For the very first time, the mysterious figure on the cloth appeared startlingly human. The expression on the face seemed calm and dignified, yet it also carried the unmistakable signs of brutal torture. What Pia witnessed that night completely transformed public perception of the shroud. Normally, when an ordinary painting or drawing is photographed into a negative, the result looks distorted and unnatural. Shadows reverse, highlights darken, and facial features appear strange or flattened. But the Shroud of Turin behaved differently. When viewed as a photographic negative, the image suddenly appeared realistic, almost like a modern black and white portrait captured from a real person standing before a camera. That discovery changed everything, because what appears on the cloth to the naked eye is already negative in nature. And that raised a question no skeptic could easily answer. How could someone living in the Middle Ages create a perfect photographic negative centuries before photography even existed? At the time, no one understood photographic inversion, light-sensitive imaging, or the science required to produce such an effect. Cameras would not be invented for another 800 years. Even more puzzling, the human brain does not naturally perceive the world in reversed negative tones. This became the first major scientific mystery surrounding the shroud. The image did not behave like paint. It behaved more like a captured moment, frozen in time. Over the decades, researchers examined the cloth using X-rays, ultraviolet analysis, infrared imaging, and laser technology. Yet scientists eventually realized that the greatest clues were not hidden in the image itself. They were hidden in the dust. Deep within the microscopic spaces between the linen fibers were tiny particles trapped over centuries. Pollen, dirt, biological residue, and fragments invisible to the naked eye. In 2015, researchers from the University of Padua, led by Professor Gianni Barcaccia, were granted rare scientific access to study the relic on a microscopic level. Their goal was both ambitious and controversial: to recover and analyze traces of DNA. The scientists were not searching for the DNA of God. Science has no reference point for something like that. Instead, they hoped to reconstruct the history of the cloth itself, identifying where it may have traveled, who may have touched it, and what biological traces had accumulated over centuries. Using specialized sterile micro-vacuum instruments equipped with ultrafine filters, the team carefully extracted microscopic material from deep within the weave of the linen. They collected particles of dust, pollen, and organic residue preserved inside the threads for hundreds, possibly thousands, of years. Researchers also studied materials saved from the 1988 restoration process, which had been preserved under archival conditions. The investigation pushed forensic science to its limits. Even the smallest contamination, a breath, a skin cell, or a stray particle introduced by priests, scientists, or visitors over the centuries, had the potential to alter the results completely. Even the slightest contamination, a breath, a stray skin particle, or a single touch from a priest, scientist, or technician, could overwrite ancient biological traces with modern genetic material. That's why every sample was transferred into an ultra-sterile laboratory environment before analysis began. Inside the clean room, scientists used advanced next-generation sequencing technology, known as NGS, to decode the material collected from the cloth. Their main focus was mitochondrial DNA from both humans and plants, and there was a critical reason for that. Unlike regular nuclear DNA, mitochondrial DNA survives far longer in ancient samples. It exists in large quantities within cells and is inherited only through the maternal line, making it an exceptionally reliable tool for tracing ancestry, migration, and geographic origin. For weeks, powerful computers processed millions of genetic fragments, comparing them with international genomic databases containing DNA profiles from populations across the globe. Then the final results appeared on the screens, and the researchers realized they were facing something far beyond a simple forgery. The cloth did not contain the genetic trace of one individual. It carried the biological footprint of humanity itself. The study, later published in the respected journal Nature Scientific Reports, triggered intense debate across the scientific world. Researchers had expected a dominant and predictable genetic pattern. If the Shroud of Turin was simply a medieval fake created in France, then most of the DNA should have originated from European populations, especially French or Italian ancestry. And if the cloth had remained exclusively in the region of Jerusalem since ancient times, the traces should have pointed overwhelmingly toward the Middle East. But that is not what the data revealed. Instead, the results look Edessa or Mandelion traveled across the ancient world for centuries. Historical accounts describe it being folded in a way that revealed only the face, displayed within a sacred frame known as the tetradiplon. Its journey is believed to have begun in Jerusalem before eventually reaching Edessa, a city positioned along one of history's greatest trade networks, the Silk Road. Merchants, travelers, diplomats, and pilgrims from China, India, Persia, Arabia, and beyond moved through the city constantly. Many came into contact with the sacred relic. Some prayed before it. Others touched its container or stood only inches away from it. And over centuries, microscopic traces accumulated. Skin cells, hair fragments, sweat particles, dust carried from distant lands. Layer after layer, century after century, the cloth absorbed the invisible biological residue of countless human encounters. The relic later traveled to Constantinople, where it remained during the height of the Byzantine Empire. At the time, Constantinople was one of the largest and most cosmopolitan cities on Earth, a crossroads where cultures from Europe, Asia, and Africa converged. Then came the Fourth Crusade in 1204. The city was violently sacked, and amid the chaos, the relic disappeared. Historical records suggest it later surfaced in Athens before eventually appearing in France during the 14th century, in the possession of the knight Geoffroy de Charny. And this is where the medieval forgery theory begins to collapse under its own weight. A European artist living centuries ago could never have predicted modern genetic science, nor could he realistically gather biological traces from China, India, Africa, and the Middle East, then distribute them onto fabric in a way capable of deceiving forensic investigators hundreds of years in the future. What scientists found on the shroud does not resemble random contamination. It resembles a biological timeline, a microscopic archive created through centuries of human contact across continents. And the DNA evidence was only part of the mystery. The next clues came not from human biology, but from plants. The science of pollen analysis, known as palynology, opened an entirely new chapter in the investigation. Two respected researchers approached the mystery independently. Israeli botanist Avinoam Danin from the Hebrew University of Jerusalem and Swiss forensic scientist Max Frey. Working independently, both researchers studied microscopic pollen grains trapped deep within the fibers of the linen. Using adhesive sampling techniques, they carefully removed tiny particles preserved on the cloth's surface. What they uncovered stunned the scientific and botanical communities. A total of 58 different plant species were identified on the Shroud of Turin. 17 of those species came from Europe, exactly what historians would expect considering the relic's well-documented presence across Europe over the last several centuries. But the remaining pollen told a far more mysterious story. Scientists identified plant species native to the Middle East, Turkey, and the Anatolian region, perfectly matching the ancient travel route historically associated with the relic through Edessa and Constantinople. Then came the most extraordinary discovery of all. Several pollen samples belonged to plants that grow only within a narrow region between Jerusalem and Jericho. One species stood out above all the others, Gundalia tourneforti, a thorn-covered desert plant related to thistles. Its pollen appeared in astonishing quantities, making up nearly half of all pollen traces discovered on the cloth. That level of concentration was highly unusual. Why would a thorny desert plant dominate the biological traces on a burial cloth? The answer became deeply unsettling when historians revisited the biblical descriptions of Christ's crucifixion, the crown of thorns. With its long, rigid spikes, Gundalia tourneforti closely resembles the type of plant Roman soldiers could have twisted into a mock crown placed upon the head of the King of the Jews. Suddenly, the pollen itself seemed to tell a story. Even more remarkable, this plant blooms around Jerusalem during a very specific season, early spring, precisely during the time of Passover. Researchers noted that large concentrations of this pollen appeared especially around the head and shoulder regions of the cloth, strengthening the theory that the body wrapped within it had direct contact with thorn-bearing plants. Another major clue came from a plant called Zygophyllum dumosum. This species grows almost exclusively within the Judean desert and parts of the Sinai region. Yet its pollen was also discovered embedded within the linen. For skeptics, this created a serious problem. A medieval artist in France could paint an image, but pollen cannot be painted. Microscopic plant traces act like invisible geographic fingerprints. They settle naturally and remain trapped within fabric over time. Reproducing such a complex botanical signature intentionally would have been virtually impossible centuries ago. For years, critics argued that the reddish stains visible on the cloth were simply pigments, substances like ochre or red paint applied by a medieval forger. That theory suffered a major blow in 2017. Italian researchers led by Professor Giulio Fanti from the University of Padua, together with medical specialists from Trieste, examined the stains using advanced transmission electron microscopy and Raman spectroscopy. At the microscopic level, they did not find paint. They found blood, human blood, classified as type A-B. Interestingly, the same blood type has reportedly appeared on several other ancient Christian relics, including the Sudarium of Oviedo. But the most disturbing discovery was not the blood type itself. It was the condition of the blood. Scientists detected nanoparticles of creatinine and ferritin attached to hemoglobin, biochemical markers associated with catastrophic physical trauma. These substances appear in extreme concentrations only when the human body undergoes severe suffering, massive muscle damage, dehydration, and shock. In cases of prolonged torture, muscles begin breaking down in a process called rhabdomyolysis, flooding the bloodstream with abnormal chemical compounds. And according to the researchers, the blood on the cloth showed exactly those signatures. In other words, this was not the blood of someone who died peacefully. It reflected the physiology of a person subjected to extreme violence before death. The findings closely matched historical descriptions of Roman scourging, brutal beatings carried out using leather whips weighted with metal fragments. Researchers identified wound patterns consistent with over 100 separate blows across the body. And unlike paint, no medieval artist could reproduce the microscopic biochemical profile of severe trauma, organ stress, and hypovolemic shock. Another mystery involved the unusual color of the bloodstains. Normally, ancient blood darkens over time, turning brown or almost black. Yet many stains on the shroud remained reddish. Scientists later discovered elevated levels of bilirubin, a chemical released during extreme physical stress and trauma. High bilirubin concentrations can preserve the reddish appearance of blood for centuries. In other words, the chemistry of the stains reflected the biological condition of a brutally tortured body. Naturally, this leads to another major question. What about the famous carbon dating tests? In 1988, 3 leading laboratories, Oxford, Zurich, and Arizona, performed radiocarbon dating on the cloth. Their conclusion appeared definitive. The fabric dated between 1260 and 1390 AD. For many people, the debate seemed over. The shroud was declared medieval. Even the church responded cautiously, referring to it more as a devotional icon than confirmed relic. But scientific understanding continued evolving. Decades later, researchers began uncovering a critical flaw in the original testing process. The issue was not necessarily the dating technology itself. It was the sample chosen for testing. The piece removed in 1988 came from the outer edge of the cloth, an area heavily handled over centuries during exhibitions and ceremonies. That corner had absorbed contamination from human touch, candle smoke, oils, bacteria, and environmental exposure, unlike almost any other part of the relic. Then chemist Ray Rogers from Los Alamos made an even more damaging discovery. The tested section had likely undergone medieval repair work. According to Rogers, restorers had woven newer cotton fibers into the damaged corner, using a technique designed to make repairs nearly invisible. Chemical analysis later revealed traces of cotton within the tested sample, despite the main body of the shroud being composed entirely of linen. Researchers also detected dye compounds and gum arabic used to blend the repaired threads with the older fabric. In simple terms, the laboratories may not have dated the original cloth at all. They may have dated a repaired medieval patch. That realization dramatically weakened the certainty surrounding the 1988 results. Critics argued that testing the most contaminated and heavily restored corner of the artifact violated basic archaeological procedure. It would be like trying to determine the age of an ancient statue by analyzing modern material stuck to its surface. As doubts surrounding the carbon dating grew, scientists searched for new techniques capable of bypassing contamination altogether. Then, in 2022, Italian physicist Liberato De Caro from the Institute of Crystallography in Bari introduced a completely different approach. One designed to analyze the aging of the linen fibers themselves rather than relying on conventional radiocarbon contamination-sensitive testing. The breakthrough came through a scientific process known as WAXS, a method that completely reshaped the debate surrounding the Shroud of Turin. Instead of analyzing contamination on the cloth, this technique examined the linen fibers themselves at the atomic level. Inside linen exists cellulose, a material that slowly changes as centuries pass. Over time, its molecular chains weaken, its crystalline structure deteriorates, and the fabric records the effects of humidity, radiation, and temperature like a natural internal clock. Italian physicist Liberato De Caro compared fibers from the shroud with ancient textiles whose ages are already firmly established, including linen from Egyptian mummies, dating back thousands of years, and fabrics from medieval Europe. The results were astonishing. The molecular aging pattern of the shroud did not resemble medieval cloth at all. It appeared dramatically older. Even more surprising, the linen closely matched first-century textiles recovered from the fortress of Masada in Israel, a site destroyed in 74 AD. Those fabrics date directly to the time period traditionally associated with Jesus Christ. In other words, modern X-ray analysis pushed the possible origin of the shroud back more than 1,000 years earlier than the controversial 1988 carbon dating suggested. Decades ago, science seemed to close the case. Now, modern science reopened it. But perhaps the greatest mystery still remained unanswered. How was the image created? Researchers found no paint, no ink, no brush strokes. The image exists only on the extreme surface of the linen fibers, roughly 200 nanometers deep, thinner than a human hair by hundreds of times. Remove the outer layer, and the image disappears instantly. This was not pigment. It was a microscopic chemical alteration caused by oxidation and dehydration, almost like a controlled scorch mark produced by an unknown source of energy. Scientists attempted countless experiments to reproduce it. They tested heat, acids, radiation, chemical reactions. Nothing worked convincingly. The closest result came from extremely brief bursts of vacuum ultraviolet radiation, technology that did not exist in the ancient world. To imprint an image across an entire human-sized cloth, researchers calculated that an enormous pulse of energy would have been required, released in less than a billionth of a second, enough energy to mark the linen, but somehow not burn it. Then, another astonishing discovery emerged. In 1976, image analyzers originally developed for NASA revealed that the shroud does not behave like an ordinary photograph. It contains three-dimensional information. The shading intensity across the image corresponds precisely to the distance between the cloth and the body it covered, creating a mathematically accurate 3D structure. No known medieval painting possesses that property. And according to some digital studies, faint shapes resembling ancient coins appeared over the eyes of the figure, objects similar to coins issued under Pontius Pilate around 29 AD. For supporters of authenticity, discoveries like these deepened the mystery even further. Combined with pollen from Jerusalem region plants, dust linked to the Middle East, blood chemistry indicating severe torture, and anatomically accurate crucifixion wounds through the wrists rather than the palms, the evidence seemed to point repeatedly toward one historical setting: Jerusalem, 1st century, around 30 to 33 AD. Layer by layer, field by field, biology, chemistry, archaeology, physics, forensic medicine, geology, and history. The shroud continued, resisting simple explanation. It did not behave like a painting. It appeared more like a physical imprint left by a real human body at a single moment in time. Today, the shroud rests silently within its protected chamber. But the scientific data surrounding it continues to fuel debate across the world. For some, it remains a medieval masterpiece. For others, it stands as powerful evidence connected to one of history's most important events. And perhaps the greatest mystery is this: if the image truly resulted from an unknown burst of energy, what exactly happened in that tomb nearly 2,000 years ago? Maybe future science will eventually provide an answer. Or maybe some mysteries are destined to remain suspended between evidence and faith forever.